I'm Veronica Miller, and I'm the director of the Forum for Collaborative Research, which has 6 sub-forums underneath it, including the PSC Forum, and they all follow the same model. And I am also on faculty at the University of California School of Public Health at Berkeley. Yes, so I started off as a basic researcher and went into clinical research and became really interested in the whole point of how do new drugs get tested, developed, approved, and then implemented, and what kind of research takes you to that point. So my most recent work with the Forum has included HIV, hepatitis C, hepatitis B, CMV, fatty liver disease, and the most recent project is PSC. And what we aim to do is to bring all of the scientists together from all the different stakeholders to come to consensus and agreement and at least a common understanding of what it takes to develop new diagnostics and new drugs and establish enough collaboration that we can make the whole system more efficient. I think HIV was the field where this patient, industry, government ability to talk to each other first happened because of the unique patient activism that happened in HIV. But that really has informed all of drug development, and it's extremely rewarding to see what's happening in the PSC community and the strong voices that patients and their caregivers have, because that's in the end really what it's all about. And the sooner they can have an impact and a voice in all of these processes, the better. It's a rare disease, and many doctors haven't heard about it. They confuse it with PBC. If I say we're working on a rare liver disease, they always say PBC. I say no, PSC, and they say PSC. And then, here I am, a virologist and immunologist by training, explaining to a gastroenterologist what PSC is. But sometimes, many of them do know. I'm not trying to disparage the field, but it is a very unique disease with extreme challenges. And we're here to help solve some of the problems. The main stakeholder that we need at the table are the regulators, because they're the ones that will make decisions as to what it takes to approve a drug. And they often start with the black box, where we don't know what the appropriate endpoint is, and we don't know how to select patients to really recruit them into clinical trials. So they need help, and they really appreciate help in the right way. And so they're a very, very important stakeholder group. The other group are the industry, simply because they're the ones that are going to manufacture the drugs and produce the diagnostic kits. But then you need the academic researchers who are going to carry out the research. And you really need the patient voice to say, this is really what we need, and this is our perspective on what risk we're willing to undergo, and what benefit we expect, and when is the benefit sufficient, and when are we, at the beginning, we might be willing to accept a little bit of benefit with a fair amount of risk just because we don't have anything. But once we have drugs that work, then of course we won't accept just a little bit of benefit with a whole lot of risk. Once we have a few drugs, then we want drugs that are better, safer, more effective. But we need to start somewhere. So this whole equation of when is benefit risk the right balance is not a static concept. And so it needs constant revision, and everybody needs to have an input. And the FDA, in their last reiteration of the Act, the 2012 FDASIA and now the 21st Century Cure Act, really stresses that the patient voice needs to be included much more consistently and systematically. So we provide one mechanism of doing that. I attended a meeting because we already were active in fatty liver disease, and it has many of the same players in terms of the companies and some of the hepatologists that are involved. And so I was invited to speak at an FDA-sponsored meeting where I talked about the liver forum model. And then basically PSC came to us and basically said, could you work in our field specifically? It's basically word of mouth. We don't sort of put a thing out there saying we're looking for new disease, but it's usually once people hear of what we do and how we do it, and if all the cards fall in the right places, then we say, yes, we can start this project. Once Hep C was cured, many of them are now working on hepatitis B. We have treatments, but we don't have a curative strategy. So many of the more antiviral focused companies are now looking at hepatitis B as the next challenge, but many of them, and I see many of the people I knew from my HIV days that are now working on fatty liver disease, and they will also, the more awareness we have about PSC, the more they will also join this field, I'm sure. But if there's a glimmer of clarity about the regulatory path, that just really helps a lot. Because it's very difficult for a company to say we're going to invest in something that we have no idea how we're going to test it, right? You have to have some idea of what the path is going to be before you make a commitment to the disease area. So that's what we try and do. We also know what the regulators are going to ask for.
Okay.
Because that's the crucial question. What kind of study are you going to do, and how will you determine that the drug is effective? And that is the hardest part, because if you always start off with the hard clinical endpoints, but from diagnosis to endpoint, it might be 8 years, 10 years, 13 years, even longer, right? And so how can you do a study in that length of time? You need to know within 2 years or 3 years if the drug is effective. So how are we going to measure that? And that's where we all need to put our heads together and figure that out. Because it's very difficult to do clinical trials that last 8 years or 10 years or even longer. So it would be very difficult for a sponsor to make a major investment if that is what is called for. But if we can find out what do we know about the disease process, that if we look 3 years or 2 years or 1 year after treatment, that will give us an indication that this drug is doing something that will actually prevent what we know might happen 8 years down the road, right? I haven't seen those results. I've seen the results for NASH, which were presented at EASL. I have not seen the results. And so basically they will learn from this trial and from the results, and that will then determine what the next step will be. Do they need to find the right dose, or have they already hit on the right dose? So they start usually with a few patients, and then they increase the number of patients, and then from there they will then do larger studies to confirm. So you basically go from the first time it gets actually tested in patients with the disease to the proof of concept that there's actually something happening. Depending on how the drug acts, what you look for at that stage is, can you actually see that what the drug is supposed to do is actually happening? So that's the first thing, that the drug gets absorbed and it does something that it's supposed to do. And if it does indeed do that, then you can go and do the dose finding and test for safety and do other things that you do going along. But then the hardest part is to say that what the drug is supposed to do actually has a relationship to 8 years or 10 years down the road. That's the hard. And that's why NGM is also working with us on these issues. So we have NGM, we have Gilead, Intercept, and Gilead, and NGM, and a few other companies that are very much involved in this. And they come in as scientists. We don't bring in the marketing people. We bring in the scientists, like the ones that are actually running the studies and determining the studies. And they sit together with people like Keith Linder and Chris Bolos, who we heard today. And we all sit around the table, and the FDA can ask questions, and we can all talk about it. And then eventually, out of all of these conversations, and we keep looking at the data and keep looking at the data from different perspectives and see how the science is actually evolving and what that means to how clinical trials should be done, and hopefully by everyone sitting in the room so that everyone can learn from the NGM experience without putting them at risk for losing IP. That's not part of what we're asking them to do. We're just asking them to discuss with us what their challenges have been and how they've resolved some of them, and also what can we learn from their science and from Intercept science and from Gilead science, etc. So that's the process. Well, first of all, the stem cell person, Natash, he's already agreed that he will be part of this effort. So we will bring him in these discussions, because those are different people at the FDA that look at those kinds of issues. And then in terms of the fecal transfer, I thought, like they say, they view it as a biologic. And it is, it's got to be the hardest thing to standardize, because you're not producing it in a lab like you do, like some of these biologics for rheumatoid arthritis, where every lot is the same, or a vaccine where every lot is the same. So I really thought we should really, at some point in the process, bring those FDA people in as well, and really talk about, okay, so how would you actually standardize it? And the whole thing is, on the one hand, so ridiculous, and on the other hand,
the numbers don't lie.
Right, yeah, right. And so, it's ridiculous about how it's being viewed, but on the other hand, you don't want to just have people doing these procedures at home, ad nauseum. That's sorry, you might want to cut that part out.
No, we want to keep it.
But there's safety issues there, and so there needs to be. But it's just such an amazing process, a procedure. And it's just kind of, you just want to laugh, right? Because it's a biologic. And it is a biologic, but it's organic, it's all of it, right? But it's full of bugs. And, you're doing something with that. And so I find it extremely, extremely fascinating. And I hope that we can find some method or some process whereby the process about how we validate it becomes easier. And like that whole story about patient B or donor B, right? That basically they said the study was a failure, but then you looked at everyone who got donor B did well. And so what does that tell us? And how do we find more donor Bs, right? That's why there needs to be research and some oversight on this. Because there could be other donors who might be the opposite of donor B. It's not that it doesn't help you, but it might actually make things worse, right? So I totally agree with them. People shouldn't just start doing it at home. But on the other hand, it's a biologic that has a very, they have all of these rules about how these manufacturing sites are regulated and the oversight, and they go in and inspect them. And how will you do that with a human donor? It's funny in a way, but on the other hand, it's so serious, because it could be so helpful if we figure out how to do it.
And then affecting so many diseases, it looks like.
Right. No, I say the gut is the new soul. I firmly believe that. The gut is the new soul, right? Never mind the brain and all of that. It's your gut. I'm just overwhelmed by the spirit and by this. I sat there at the table and I feel already like I'm a family. Although I don't have anyone in my family with PSC and I don't have PSC, but there's something about the magic of this community that is extremely, extremely powerful.